inaugural Science Yak podcast, where we'll be talking a bit about science, a bit about news, and just hopefully entertaining you all a little bit. And when I say all, I'm referring to the possibly three of you listening to this. I'm Matt.
And I'm Thomas. Yeah, so hopefully you all will be enjoying spending a little bit of time with us. Maybe we'll be going off on a bit of a tangent, having a little bit of conversation. Should be a bit of fun for everyone. The first little piece of science news that I'd like to refer to today is something that I saw in many news articles, published very recently, referred to often as a Queensland cancer test. So the headlines went something to the effect of, the discovery of a unique DNA signature common to multiple cancers could one day revolutionize the way we diagnose cancer, particularly in its early stages, Australian researchers say. Well, it's a pretty big headline, and I think it's important to look into some of those claims.
So were they, in the article, were they claiming that they had a special material that did this, or an actual mechanism to do it?
So I looked through the article, and I found that it was a little bit vague because it is made for layman's people to understand. So I actually delved into the actual journal article published in Nature Communications, which, in case you didn't know, as I didn't, that's an open source part of the journal Nature.
Do you have the issue?
No, I do not have the issue number with me at the moment.
Just in case people want to look at it themselves.
However, I did look through it for myself, and you can find it if you go to the ABC article, links to it in the sources. But basically what they actually did, these researchers, is that they discovered that there is something which is known as an epigenetic methylation landscape. So, in case you didn't know...
For us that don't do biology.
Yeah, for those of us who aren't familiar with biology, well, which is most of us here, in fact all of us, so you're probably aware of what the genome is, the DNA, the base pairs, adenine, cytosine, guanine, thymine, etc., etc. But the epigenome is actually what determines what genes are activated.
So, if I remember this correctly from my brief reading of some biology stuff, epigenetics relates to signal proteins that are actually attached to the DNA, the genome itself.
Yeah, so I'm not an expert in this field, but it basically determines how certain genes are activated. So the coding is all in there, but there's also like subcoding that determines what is expressed within the organism. The methylation landscape describes the way that methyl groups are attached to the genome. Specifically, they're attached to the cytosine base, making methylcytosine. You don't have to worry about the names, though. The point is that the researchers discovered that you could determine the nature of the genome, or the nature of the epigenome, through measuring this methylation landscape, and that there was a detectable difference between those the epigenome of cancer cells and those of normal cells.
So, going back to the methylation landscape, was it? Correct. So how were they able to measure that inside the cell, or how did that relate to it?
So what they actually do is they don't measure it within the cell. They take the genetic material from the cell. So they take the genetic material out of the cell, and they do what is basically an adsorption test. An adsorption test is essentially a test for how well material adheres to a substance. So I hadn't heard about this before, but certain materials will adhere to certain levels, and that can be measured.
So I'm imagining this case as someone who studied chemistry. The absorption, the methylation landscape, they're going to measure how much it sticks due to the hydrogen bonding that occurs, I'm guessing.
Funnily enough, I thought that it would come down to intermolecular forces, but when I looked into the journal article, the specific way in which methylation landscapes interact with the surfaces here is not yet known.
So before we scare the three of you off, just to clarify some of this stuff, when we're talking about intermolecular forces, we're just talking about secondary forces that occur between molecules. So you have forces like covalent bonding and ionic bonding, which hold atoms together to form molecules. And between those, you can get secondary forces that just slightly negative or positive areas of molecules will hold each other together. That's why water stuff can dissolve in water, because the hydrogens on water molecules are slightly positive and the oxygen is slightly negative. So any molecule that has a slight polarization like that as well can then will be attracted to those molecules and actually bond into the water and dissolve. So hopefully that just gives you a bit more hint. As such, materials that have those sort of properties can actually will actually stick to each other more, and so talking to Tom about the, what was it, absorbance? Adsorption, of measuring the adsorption. That I would have thought that related to secondary forces because those would help groups of molecules stay connected together. So what was it actually?
Well, I was just saying how that essentially this is not an area which is yet understood, because the adsorption we're looking at is actually requiring some very recent developed technologies, some very precise equipment. So the way they measure adsorption is that they essentially use a gold sheet, a very finely flat surface of gold, and then they allow the material to adhere to it after a certain determined amount of time, and then after that they measure an electric current through it.
It's amazing how much gold comes up in biology, because wasn't that originally how they tried to do genetic engineering, was just attach ionized DNA to gold particles and then just fire it into an organism? Well, specifically plants. They weren't doing this to animals.
I'm not sure if that is the first way that they were doing it. I understand that the random mutation systems which they had were one of the earlier mechanisms. However, it's true that gold actually is one of the metals which most organisms, including humans, can consume without being in any danger of heavy metal poisoning, like lead or any other particular things. We've just found out something called the gene gun.
So, good old Wikipedia. Just to make sure I'm not talking like a crazy person, the gold used in something called a gene gun or biolistic particle delivery system. And I'm saying biolistic, not ballistic, which I think they meant as a pun. So what it seems to do is they'd actually use heavy metals as a payload to attach DNA plasmids to fire into an organism. Let me see if I can find a diagram of that, because that sounds hilarious. Yep, so it's pretty much direct delivery of DNA into cells by shooting metals at it.
Well, there you go. We are all learning something. The application of gold in this sense is actually just because we can as you know, gold has a very, very high conductivity, and so we can very finely measure the differences in adherence of the genetic material, which I was referring to. Continuing on, the methylation landscape that determines the adherence. So this usually has a parabolic relationship with the methylation of the genetic material. So the methylation is actually just how many cytosines on the genetic material have become methyl cytosine. And it essentially has a parabolic relationship with a maxima sort of halfway between 0% and 100% methylated. Normal human...
As the number of methylated cytosine molecules, was it? Or cytosine. Bases. As they become more methylated, you expect to see the attraction increase parabolically.
I don't think it's quite accurate to call it attraction. It is adsorption. So it is, yes. And the theoretical reason for this is actually because of another secondary force, which is the aggregation. So as the methylation increases, the attraction to adsorb does increase. However, also there is a greater willingness for the genetic material to aggregate, so it forms these clumps, and as that level increases, it's more likely to form clumps instead of adsorb correctly to the material. The most interesting thing that the Fuddy study found, however, was that the way that cancer cells developed was not a sort of uniform demethylation, as you might expect, or some sort of a uniform effect. They found that after the difference between a normal cell and a cancer cell wasn't just in the total methylation level. The cancer cells actually had what was known as methylated clusters. So what you actually found was that there was these small clusters of methylated bases separated by long intergenic lengths. So that's just along the genome polymer that didn't have that, and that this actually led to a disproportionately higher adsorption of the genetic material. Interestingly, though, this wasn't the only application of this technology, because as you might understand, not every lab can have some extremely high quality adsorption measuring equipment. I'm not sure if you remember colorimetry.
Yeah.
So colorimetry is basically just measuring I'll let Matt explain colorimetry.
So yeah, colorimetry is a qualitative analysis technique where... Wait, what are you talking about?
It's not entirely qualitative.
Which maybe you should go back to this because I think I might be thinking of the wrong one.
So colorimetry basically is you measure the amount of of certain wavelength of light being absorbed or released by a solution material. And so you might be wondering, well, how does this apply to yeah, yeah, it's chromatography. Not quite the same. That's the one with the Skittles and the M&Ms, I believe, with the milk.
Well, poisonous, what was it? Toxic materials inside of, toxic dye inside of, what was the drink?
Oh, like 7.
Cordials.
It was cordials.
Yeah.
No, in this case we're talking about colorimetry. And so what they found was, I don't know if do you know what colloidal gold is?
No.
Okay, it's basically...
I can take a guess, but I'm not sure how it would be. So my guess on what colloidal gold might be is just gold that is not dissolved in a solution, but still present.
Yeah, so the key part about colloidal gold is that you basically have these floating, this mixture of very fine gold particles, or as they're often described in the articles, nanoparticles. The key point... Oh, yes?
Sorry, you were about to say it, but the key point with that being the distinction is when something dissolves, it's actually a chemical process, whereas in a colloidal suspension, they're not actually dissolved in the solution.
That comes from the fact that gold is, of course, significantly difficult to dissolve, as aqua regia does the trick, but aqua regia is also incredibly dangerous. Yeah.
Probably destroy part of the genome if you tried using that, right?
Yes, that would not be good. But basically the point is that this colloidal gold can be reacted with salts. So you introduce the salt and you basically get a change in color.
Yeah.
So that goes along with, all your classic like drug testing, if you imagine, where like something turns blue, etc., etc. But what happens in this case is they can mix the colloidal gold mixture with the genetic material. And so when they mix it with the genetic material forms what you might think of like a mossy barrier along the gold. So you can guess what happens when the salt is reintroduced into that solution. Is that in that mixture?
So you're measuring the concentration of the gold left over after you observe the color change. So you're measuring the intensity of the color change to know how much gold is left.
Yes. Well, the gold is still there. The gold never leaves. The point is it's actually how much gold has interacted with the salt that's introduced. However, of course, if you have a highly adsorbed genetic material, the material's been able to adhere to the gold very well over that period of time, the salt does not interact. And so you can actually find, and they found it was with roughly 75% accuracy, which was less than using the electrical measurements. They could determine whether its genetic material was cancerous or non-cancerous. Worthwhile of note, though, that this was not for just any or all cancers. As with any study, they were limited in what they had access to. This one studied with breast, prostate, lung, and colorectal cells, and it found this difference between normal and cancerous cells in all of those cancers.
That's the interesting point, because the big problem with cancer is it's not just one thing. It is something that is individually treated, sometimes on a patient-by-patient basis, not just a type of cancer-by-type of cancer basis, which makes it really hard to fight, because sometimes solutions need to be highly specialized for any given type of cancer.
Yeah, so many people have misconceptions about the fight against cancer. They sort of think that we're going to find like a golden bullet that will allow us to just defeat all cancers. This is not a very realistic or likely sense, because if you know how cancers develop, every cancer can develop, well, essentially does develop independently of one another. So they all have different genetic strings which allow them to turn into cancers. So it's not likely that we're ever going to find one golden bullet that will stop these cancer issues.
So one of the more recent breakthroughs, which we were actually fortunate enough to go and get talked to about, was at the Weehai Medical Facility. That's down in Melbourne, Australia. And what they developed, this is a recent breakthrough, I think it was only this year it was announced. They actually have found a specific drug that can attack, which was it, lymph node cancers, or cancer?
Probably likely lymphomatic.
Yeah, but it actually was able to cause the cancer cells to undergo apoptosis, which is cell death. So amazingly, this drug could actually link on to specific cancer cells and cause them to self-destruct. that's actually one of the big reasons that some cancers exist in the first place is the genetic information that tells a cell to commit suicide after a certain amount of time doesn't function, so it just continues to reproduce uncontrollably and it doesn't die itself. And that is what the buildup of cells in tissue is what causes all the problems that cancer is associated with.
Yeah, and it's interesting that you mentioned that, because that sort of thing, like not being able to undergo apoptosis as well as unregulated cell division, are something that are common to all cancers. And so, as is shown by the study with the colloidal gold, we should expect that there should be biomarkers, epigenetic biomarkers, in fact, common to all cancer cells, which should assist in identification. And hopefully that's something we're moving towards. Ultimately, however, the treatment of each cancer is going to come differently depending on where the cancer is located, what kind of cancer it is. And so we should expect that the drugs will be tailored to, or the drugs or treatments will be tailored to each and every individual case.
Yeah, so that pretty interesting. What did ABC report? Did you think anything was a bit lacking in their report?
ABC did a pretty good job. They didn't make too many big claims. It's interesting that they state that it is particularly good at diagnosis in early stages in the headline. When I delved into the study, I actually found that the thing is that not all genetic material expresses the cancerous alleles, so you shouldn't expect that they should all have the methylation landscape. And so they did tests where they reduced the amount of genetic material which was expressing a cancer methylation landscape to 10% and lower, and they found that they could still discern difference except for up down to less than 1%. And so for this reason, they said that it likely wouldn't be useful for detecting in early stages. So when they said it's capable of detect in early stages, maybe if you're using the electrical measurement method. However, that's not the likely common use method, well, at least for the time being. So I don't think that was quite entirely accurate in their reporting. But other than that, they strayed away from making any big overarching claims. The study was in essence exploratory. It's not something that had a massive sample pool. It was a significant sample pool, and the claims are made with like 90%, 95% confidence. But we can expect that in future there will be larger trials with more people, and maybe even twin-controlled trials.
Talking about twin-controlled trials, now those are some really interesting ones. Now, I have to imagine, is there some sort of medical registry for twins? Because that's something that is extremely useful to studying something. A famous case, I think, was actually studying one of the big questions always is nature versus nurture when raising children. And that's the classic case for a twin study, is if you take twins who should be genetically mostly identical and separate them, how different can they actually turn out? And I don't know the results of that one, so I won't comment on it. But one of my favorite examples of a twin study was actually one that NASA did, sending with astronaut someone Kelly, was it? Tom Kelly? Tom Kelly? I need to double check.
I'll look it up.
Yep. So anyway, to see better understand the effects of space travel, two identical twin brothers. That only finished last year, didn't it? His Command in Space. It wasn't John Kelly, was it? Or...
Just give me a second. I'll have the data.
No, it was
Mark and Scott Kelly. Mark and Scott Kelly were the two astronauts.
Right. So it was Scott Kelly went into space, didn't he?
It looks like that's the case.
So they wanted to better understand the effects on DNA, bone loss, tissue regeneration, and all that space travel has, because there are a lot of still unknown problems with space travel. Talking from a physics standpoint, which is my preferred standpoint, one of the big issues is going into the upper atmosphere. Means that a lot of the shielding from solar radiation and cosmic rays is lost. So astronauts who usually go up for a couple of months at a time, 6 months, sometimes a year or so, will experience more radiation than most of us will for our lives. We still experience radiation here on Earth, but significantly lower doses. And one of the big questions is what kind of effect can this radiation have on biological development and stuff. So that's a question I'm worried about. Especially when you're living in a metal capsule, because one of the problems that you can have in a metal capsule when you're being hit by ionizing radiation is you can actually get secondary radiation occurring, where a high energy alpha particle, which is a helium nucleus, can just ram into a wall and eject tons of beta particles, electrons, from a metal sheet because they're all just free floating. That can be more dangerous sometimes than if you were just exposed to the alpha particles to begin with. But that's actually probably one of the more minor issues they're looking at, because they're more concerned with bone degeneration, tissue degeneration, cornea, lenses in eyes and stuff is a big problem. And the fantastic thing about a twin study is they should be they should be identical beforehand, so any differences afterhand should be relatively easily pointed out.
It's funny that how we were just talking about epigenetics, which, as you would expect, will not be the same between two twins. So genetics is a big variable, and the reason that twin studies are so popular is because it's essentially the biggest factor that affects your biology growing up, apart from nurture. So it is a very important thing to control for. But of course, as we're learning more and more, epigenetics plays a big role in our development, and that's something which is essentially impossible to be controlled unless you're fully controlling their environment as well. Just looking through the press release by NASA, there's a little interesting tidbit here that says that Scott Kelly, that is the astronaut who did go into space, had his telomeres lengthened. Now, I'll let Matt explain to you what a telomere does.
This is something I really like, even though I'm not proficient in biology compared to many other people or most other people. Telomeres are really interesting because they are actually the ends of your DNA caps. So every time your cells replicate, they have effectively little caps on the end to stop it from damaging the actual DNA. So every time your cells replicate, those little caps on the end take a bit of damage and start to degrade after a while. And that's what inevitably ends up causing aging. Is after you get to about the age of 30 or so, most of the telomeres on your cells will have degraded, and so your cells starts to the actual DNA which codes your information will start to be broken down a little bit, and that's what causes aging. And in some cancer cells, the reason they become cancerous is because they grow these very long telomeres, which means they can keep replicating over and over again without actually themselves having significantly more damage than they already would have.
Interesting that perhaps that's something in cancer cells that we could actually use ourselves. That could be, if we could discover what part of the genome enables that trait, we could in fact make a drug that allows it to happen in humans.
Well, the funny thing about that is actually that we do know of animals that have self-replicating telomeres, or not self-replicating, but cells that repair them, like the naked mole rat and lobsters. But the thing to note with them is that you'd think that they would get tons of cancers throughout their whole life because they're pretty much making cancerous cells. And the thing is, they actually do. They get a lot more cancer cells than humans would. The only difference is they have a significantly better immune system that deals with them far more efficiently.
So what you're saying is that we all need to inject ourselves with naked mole rat DNA, of course?
Absolutely. For all you superstitious people out there, that's what you do. No, don't do that. That's not how it works.
Clearly, you should follow the homeopathic method and get perhaps like 1 milliliter of naked mole rat DNA mixed in with 1 ton of water.
I'm about to flip the desk. Don't do homeopathy. It's stupid.
Worthy of note, though, is that Scott Kelly's telomeres did catch up. Essentially, they did shorten back to expected length within 2 days of his arrival on Earth. So it's a temporary occurrence. So if you're looking to live forever, perhaps going to space won't really do the trick. There are a lot of other effects, especially to his immune system. There's just, the thing is that a lot of people like Elon Musk, etc., like to talk about setting up, a Mars colony, all these large space exploration plans. But we still have a lot to deal with about the human factor in these equations.
Yeah, that's actually a great topic to talk about now. Is that space travel is actually obviously going to be really important in coming generations, because it's the last frontier we have to explore. Not actually, we still have to explore the ocean significantly, and there are a few islands which no one's been to. But one of the things with space travel is that we know so little about how it will affect astronauts, because we've only ever had them in extremely close proximity to Earth. Like a trip to Mars would expose you to even more radiation than you would be exposed to on the International Space Station. And even the Apollo astronauts only had a few days of interplanetary radiation just going to the moon. Traveling to Mars will take a few months. And that's one of the real concerns is as much as I think we're both for space travel, going to Mars, I would say it's a bit jumping the gun to go straight to Mars. So moon base, exactly. Because my thought on the idea would be is that a moon base is the perfect practice ground. You're 4 days away from Earth, which means you have the possibility of resupply. If landing goes wrong, which was a real concern of all the Apollo missions, landing on the moon is very hazardous because of the landscape itself. Parts of it are smooth and you want to aim for them, but a lot of it is very rough terrain. But yeah, so well, what were we talking about? All right, so medical concerns with space travel is something we really need to look into. Instead of just thinking we can do everything, because going to the moon, going to the Mars, a 3-month journey from which we don't know what necessarily the consequences will be, is a bit of a leap.
Yeah, I've also seen a lot of hype around the sort of what they call it, geoengineering, or the not geoengineering, the sort of climate control stuff they do in sci-fi.
Oh, sorry to take the microphone away from you again, but no, what you're talking about is just literally terraforming. One of the science fiction books which I would highly recommend people to read, by not sponsored.
Unless you're willing.
Yes, no, we do accept donations and or bribes. No, by Kim Stanley Robertson, is the Mars trilogy, first book being Red Mars, which is really interesting because they do explore in a few decades' time what it would look like if we could colonize Mars, and what the terraforming would look like if we ended up doing that. Now, for our capacity here on Earth, some of the hypothesized means of warming Mars to be more temperate for us would be to melt the polar caps, which are on Mars are not water but all CO2. And the hope would be that would create a greenhouse effect, which would help store more heat on Mars, which would lead to a better environment. I can imagine that it's not dissimilar from the environment that a primordial Earth would have seen. I can't remember what the exact numbers are, but primordial Earth, when the Earth was forming, and about a 500 million, or no, that's not it. That's too short. But I can't remember the exact times right now. But when Earth was forming, it had a really high carbon dioxide concentration, and over time, that was only reduced by one of the first forms of life to go onto the land was plants. And so I would imagine something similar would probably have to take place on Mars. Again, this is just speculation on our part, and we do not condone going straight to Mars. As much as we want to, is that you would have to, as soon as you get your warming effect from greenhouse gases, you still have a really thin atmosphere, which would be mainly CO2. So the only life that can live there then would be plant life, which means you're going to have to wait a few generations before you could actually get enough plants to Mars to replace the atmosphere with oxygen. Actually, this brings up an interesting point. I'm going to hold the microphone from Tom for just a little bit longer because I've gone on a tangent, as we warned you. The atmosphere on Mars is extremely thin. So that's probably some of you were wondering, well, how can I make the Martian atmosphere thicker with CO2 only to replace it with oxygen and still have a hot planet? The whole idea would probably be to make the atmosphere really thick and dense and bring it up to Earth. Actually, can you look up what is the atmospheric density of Mars? But coming back to Mars, terraforming Mars is probably a long way off. The energy required to melt the polar caps could probably be done by launching a lot of nuclear warheads at it, but then you're going to have to deal with the aftermath of launching nuclear warheads at the planet you want to live on.
So, just looking it up now, if you wanted a rough approximate of the Martian atmosphere, you basically would just have to cut out the bottom 35 kilometers of our own atmosphere, and what remains is the pressure and perhaps more than the volume of the Martian atmosphere.
Does that cut out the bottom?
Yeah, so you remove that bottom 35 kilometers. The at 35 kilometers, it's at the highest pressure that it reaches at Mars, so not exactly human livable conditions, perhaps. But however, of course, as Matt was explaining, terraforming by heating, that does of course play into humans' greatest strength, which appears to be at the moment involuntarily heating up planets to the point of major climactic change. I do think it is a little rather ambitious to speculate so much on this fact, considering that, like, as I was just mentioning, climate change, we're not really able to terraform Terra, Earth. So it's perhaps a little ambitious to think that in our lifetime, or even within a reasonable 200-year time frame, we can expect to see some sort of major terraforming project that succeeds on Mars. We are more likely, though, to see in our lifetime, the moon base that we were all hoping for. And perhaps then, the beauty of the moon base idea is that, of course, that it's already partway out of the gravity well. So if you can assemble a larger ship on a moon base, you're part of the way there.
So if you couldn't tell, we are very pro-moon. Just as a pragmatic option. Yes, we are the moon people. No.
Green man.
Yeah, pretty much. But as Tom was saying, the extremely beneficial thing of going to the moon is it's pretty much a jumping off point. It is a significant waste of time and energy to build every rocket you want to launch, everything you want to launch into space on Earth, because of how much infrastructure that will actually take. A far smarter option and more economic one will actually be to send parts into space, which will be a lower fuel cost because the mass of rockets will be less. So, actually, let me just take a few steps back. So, major costs of space flight is fuel and the rockets themselves. That's why the shuttle program went underway. It was to try and make a reusable space launching framework, which would turn out to be more expensive than people thought. But now that sort of technology is being followed on by SpaceX and stuff, which is highly, well, what's the best way to put it? It's great.
They do all right. They go all right.
Yeah, they're doing pretty well. But one of the things by having using the moon as a base to actually build rockets to launch off from there from is you don't need extremely large rockets to carry massive payloads, which means the actual infrastructure of the rocket boosters themselves weighs less, which means you require even less fuel. So it's all the reverse of the problem going forward, which is if you want to launch something into space, you need fuel. The more fuel you add, the more it weighs. The more it weighs, the more fuel you need to add. Going back from that, if we can reduce the weight of the rocket, we don't need as much fuel and we can make more economical trips.
So me and Matthew were at a friend's house watching Doctor Who, and I remember showing him a video by the YouTube channel Kurzgesagt - in a nutshell, which I recommend to anyone listening to watch. It's a very good science communication channel.
Yes, because we can recommend it.
However,
three of you watching, go check it out now.
What I found, what this video was about, basically this idea that the amount of junk we're putting into space, we are perhaps leading towards another kind of disaster, which is where you can essentially have a chain reaction as old satellite parts and boosters that are left in low Earth orbit collide with each other and create a chain reaction of interaction. Oh.
Just to put a quick analogy in there for any nuclear physicists, it's pretty much the large-scale version of nuclear fusion. Fission, sorry. Please correct that. Fission.
A minor mistake. There's very little difference between fusion and fission, I'm sure. But in essence, this the moon base idea is better in this aspect, because if you can have smaller rockets delivering only parts as opposed to a single rocket which has all of the things needed, you would essentially have just a giant rocket with huge amounts of fuel. You'll also be putting more and more rubbish into our atmosphere in comparison with if you're just making a shorter trip. You'd hopefully be putting less rubbish into the atmosphere. And it's just as well that we're talking about SpaceX, because they're obviously contributing in a good way to decreasing this problem, because they're pioneering this idea of reusable boosters, reusable rockets, which is, of course, like you're saying, something that NASA tried.
Yeah, well, going back to talking about this is a really important issue, and it's also a very difficult issue. So most people on Earth would agree cleaning up trash isn't too difficult unless it's floating in microplastics in the ocean. But usually, if your house is messy, you can go around and pick it up. The thing about dealing with space debris is you're in an environment now where you really have to be worried about how everything interacts and moves against itself. Because on Earth, you have the great benefit of being stuck on a massive sphere, which is so big that you can put as much momentum into it as you want. It's probably not going to move, and you can push off it, and it'll stay roughly in place. problem with space is any motion you need requires an equal and opposite momentum change to occur, which means a lot of the time maneuvering and capture procedures going on in space is significantly difficult. I don't know of any good simulators that people could use for that, but essentially it's going to makes it significantly harder to deal with space debris because of how free its motion is, and how even small changes to velocity can significantly put you out. One of the big things about space travel is, to give you an idea of how difficult it can be and how unintuitive it is, if you want to speed past an object in orbit, you actually have to decrease your thrust to get past it, because by doing that, you put yourself in a lower orbit at a higher velocity, which means you can then catch up to it, and then you raise yourself to a higher orbit where you meet up with it. So it makes it really difficult when you're dealing with space debris, which not all of it can be tracked, and it may be colliding with other stuff at the moment right now. So that gives you a sense of how difficult maneuvering can be in space, and how much detailed and calculated effort needs to go into dealing with those sort of problems.
So earlier on, Matt brought up the point of microplastics in the ocean, and I happened to just look up a few things because I'd heard a little bit of news about this fact, and so I decided to look into it. That there was this idea going around that there are bacteria that are adapting to consume these microplastics and something to that effect. And so what I found was that there are certain bacteria that are creating enzymes that can in fact break down plastic. Unfortunately, at this point, it's not a large amount of plastic that they're able to consume. But I was just looking through this other article on, I don't know how good the conversation.com is, but
just like to add one thing on that. One of the problems with microplastics in the ocean is that plastics are all, funnily enough, organic molecules, which may sound a bit strange because we all think of plastics as one of the most inorganic things to ever interact with nature. But no, going through the chemistry definitions, plastics are carbon chains, which are indeed organic molecules. Plastics mostly are polymers, long carbon chains that are joined together. In your body right now, you have long carbon chains in the form of proteins, which are pretty much what allow you to do everything. Even your DNA is itself a polymer. And the thing is, obviously we know our DNA is pretty much entirely biodegradable. We know we're pretty biodegradable. And the thing with plastics is the reason they're not biodegradable is because a lot of them are very compact. And dealing with this sort of stuff, they haven't, because of the way they're structured, they're being man-made, manufactured. They obviously don't occur in nature. So animal life and bacteria have not evolved to be able to consume microplastics. If microplastics had been around a lot longer, if they occurred naturally, it is quite likely that actually a lot of bacteria would be feeding off them as a source of energy because they are just long carbon chains. So some scientists, there's two realms of thought at the moment, which is one, it's more than two, but the two main ones are using bacteria to try and actually get them to be able to digest the plastics, and another one is to get plastics that are actually strong enough that can be used for commercial use, but have a structure similar enough to other biodegradable materials. So some universities, and I can't remember which ones they are, so I recommend you look it up because I'm too lazy to. But they are looking at actually developing plastics which can be easily consumed by bacteria we already know of at the moment.
it's actually kind of funny. This leads into another thing that I learned recently. Tangents, but a good tangent. Another YouTube channel that you should check out, Cody's Lab. He did a video making essentially a time capsule of the Carboniferous period. Carboniferous period is named so for carbon because it happens to be the period in time where almost all our coal comes from. And I didn't know, but there is a reason for this. And the reason is that it's during this period that plant life on land developed a new kind of polymer, apart from cellulose and hemicellulose, that is known as ligand. I believe it's ligand. You might want to look at the pronunciation there. But I'll refer to it as ligand. And essentially this is another kind of long chain polymer. The problem was that at this point in time, there was no life on land that could break this down. So essentially you had all of these long chain polymers that were just sitting around not being consumed If you've ever seen pictures of the Chernobyl exclusion zone where the trees are not rotting, they're simply sitting there, it might remind you of that. The lichen was then allowed to form coal because it wasn't broken up. And so that's why it's from this period that we have found most of our coal, because there was nothing to break it down to small parts that could re-enter the environment. Another one of the many reasons why coal does not work as a renewable resource, even though if you look at it in a technical sense, it is not nearly as renewable as, say, biofuel or other organic fuels. Matt's just looking into a bit of stuff about the Carboniferous period at the moment. Interestingly, that same video, he also linked to another one where someone tried to replace the polymer lichen in wood with a different material, which is actually based off of a study where they were able to achieve just that, because it turns out that is this material that gives wood its brownish color. So if you're able to remove that, you essentially end up with sort of white cellulose, except for if there's, of course, green in there for chlorophyll, allowing for photosynthesis. As I was talking about earlier with microplastics, the other kind of indigestible organic molecule, there isn't only the idea that it is going to be digested by plastics. There's also looking into how our ocean life is actually adapting to live on and live off of these new plastic environments. So obviously we scuttle ships to allow for development of coral seas. It looks like in a similar way, plant life is developing to use the niches and the protection afforded by microplastics.
So this stuff brings up another really interesting point. Another other one. But one thing that's really, or several things that are quite interesting, is that we see speciation still occurring today in a lot of areas as we humans like to slowly take over the earth. Not necessarily maliciously, but definitely not with any sort of conscience. But this brings up an interesting point, which is obviously as men of science, we boys of science, yeah. Climate change is a really big issue, one coming up much faster than we would like it to. That it is upsetting to see so many people deny it. Anyone who engages in the scientific method knows it's important to be skeptical. Anyone who denies anthropogenic climate change at this point, which just means human caused, is just a denier. And it's important to state that no one at this point is being skeptical. All the information says yes, it is human caused. And one of the arguments, people are like, oh, it's not a big deal. It's not too big a deal. We can deal with it fast. We'll deal with it quickly. It's like, well, we can't. It's the climate's not weather. It's not going to go away quickly. But one of the important things to point out is the reason we want to deal with it is can be completely self-centered. It's going to affect us in the long run. The great dying extinction event where 95% of life on Earth passed away.
Another climate scientist fear-mongering.
Oh, absolutely. No. This was Devonian? No?
Yeah.
I think Devonian period, there was what is known as the Great Dying, which is one of the largest extinction events in Earth's history, when 95% of all life on Earth died.
Triassic extinction.
Sorry, Permian Triassic, so I got that completely wrong. So that was the Permian, between the Permian and Triassic, we had the Great Dying. 95% of life on Earth died, and yet we still came back. And that's the thing, is that with life on Earth, humans will probably pass away due to climate change long before all species of life do.
Hopefully not.
Hopefully we don't at all, but continuing this way, I mean, in the long run, the Earth will probably be fine, and we won't be on it. We won't be fine. So the whole reason people want to do something about climate change is not just because it affects the ecosystem, although that is a very good reason to do something. It's also because we are the ones who it is directly going to affect, and we see that more and more as the sort of distribution of storms continually starts to spread to more and more powerful storms. So just to clarify, I'm not saying that all storms are going to become more powerful. It just means we're going to see more powerful storms more and more frequently because of rising sea temperatures.
So the way to talk about storms and climatic events in the context of global warming or climate change is that you shouldn't say that any single event, any cyclone, is caused by anthropogenic climate change. The way to say is that the general trend in the increasing number is caused by climate change. You can't say that any given event is because, of course, with the butterfly effect, it's basically impossible to know that.
Yeah. So what spurs a cyclone is a bit like asking which atom caused that dog. You're not going to know. It was a group effort. So yeah, it's a big issue, which obviously we're going to say needs to be addressed. And obviously we're just sitting here saying it needs to be addressed, not really doing much, but hopefully maybe telling the 3 people listening to this, you might tell someone else and they might agree with you and they might do something.
I understand from a purely practical point of view why it's good to bring up this economic factor of climate change. The fact is that, our modern world is kind of driven by what is economically right or wrong. But I find that as an actual moral or ethical standpoint, it's a little bit frustrating that, because there are many things in the scientific realm and in the social realm that if we were to justify it with a purely economic means would simply fall down. And that I think it's important that after we deal with this, we start to move towards the idea that ecosystems and life is worth preserving for its own sake, the same way that humans are.
This is kind of interesting. So one thing people are realizing with the climate at the moment is that it's not going to be enough just to stop and reduce emissions. It's also really important to start absorbing them back into the earth and storing them. One of the largest sinks of carbon dioxide is actually the Earth's ocean. So a problem that occurs, what happens is you get a lot carbon dioxide dissolves in water to form a carbonate molecule, which is an ion which forms carbonic acid. Problem is, as we start pumping more and more CO2 into the atmosphere, we're going to actually reach the ocean's ability to store carbon dioxide, and so we're actually going to acidify the oceans by generating carbonic acid, which will be dangerous for many reasons. One, changing the pH, which is the acidity of the ocean, greatly affects all biological systems' ability to regulate themselves. So in humans, we have pH buffers that allow us to maintain a consistent pH internally. A lot of life will have, all life has the same thing, but by raising the pH so high, especially with something like carbonate, it'll actually make it really hard for organisms to regulate their own internal processes. And just to add on to that, another thing is crustaceans are at a big risk, because crustaceans form their shells from calcium carbonate, which is solid in the water. When you dissolve carbonic acid in the water and raise the ocean's temperature, it increases the amount of calcium carbonate that can dissolve, which means instead of forming a solid, it'll actually all just stay in solution. So crustaceans won't be able to actually form shells to protect themselves.
There's kind of a cruel little irony that Matt stepped upon there, which was that he mentioned pH buffers in our blood, as well as the increasing CO2 levels in the ocean. The irony here is that, of course, the pH buffers in our blood are formed by CO2. Of course, however, our body works to keep them regulated to the right levels, whereas it doesn't seem we're doing that to the ocean.
So, just a quick chemistry side step. Carbonate is actually, diprotic buffer? Yeah. So it actually is like, for lack of my exact terminology, it is a 2-step buffer. So it can actually keep pH consistent around some specific areas. Which is a problem if we're going to saturate the ocean with it. I was going to go on to something else after that. All right. But talking about carbon sinks and stuff, the ocean being the largest one, which we are currently reaching saturation. Oh, no, sorry, I shouldn't say that. We are going to saturate, and it's going to become impossible to dissolve any more with it, which will increase our emissions problem exponentially. It's important that we start having other sources that can actually drain emissions from the atmosphere. A recent study which I heard about was the use of volcanic ash on plants as a fertilizer, which actually showed an increase in CO2 consumption. I believe, and I couldn't find the exact article again. Now, I think the report, it's a preliminary report, so it's just starting its research. If you're interested, do look it up. Although there will be articles, if you look up volcanoes and climate change, there will be articles of people saying volcanoes cause climate change, not humans. That is not true. Volcanoes emit far less CO2 than humans actually do. And I wish I had the actual numbers with me. And I do not have them on hand. So do look it up for yourself. And check, use reputable sources, Britannica and stuff. I don't know if Britannica will have anything on that. But one of the sources of carbon consumption people are looking at is plant life, as that's the thing that consumes all our CO2 at the moment. Increasing the storage capacity of that would be helpful in many ways. One, it removes CO2 from the atmosphere, and two, we can help diversify ecosystems and actually increase rehabilitation of some ecosystems by allowing for plant growth.
So there is something that I do see often brought up about the plant growth idea, is that we should move towards plants which have a higher rate of growth, which more quickly suck out CO2 from the air, which is in principle good, but it's important that, of course, ecosystems are very complex, so it's important that we don't try and support the development of certain plants in areas that aren't correct, such as many people will say that we should work towards, instead of using bamboo, we should start instead using bamboo, which grows much more quickly, and we can use the byproduct to make paper, etc., etc. This is a nice idea, but the problem is that bamboo is actually incredibly nutrient intensive. So if you're using the wrong plants in the wrong area, you can drain a soil of all of its nutrients very quickly. Whereas if you're using a tree, it creates leaves, which will eventually form soil and give back to the earth.
So yeah, you bring up a really important point. I just want to clarify as well, though, that we're not saying bamboo is not a good substitute for some forms of material. In regulated circumstances, it is. I am currently wearing socks made of bamboo. I don't know the brand name. Bamboo is really good for paper production because of how fast it can be made. It's good for some fabric production. And I don't know how they turn into fabrics, but it's very comfortable. We want sponsors. But one of the things is with bamboo, it's always, as Tom said, really nutrient intensive, which means you can starve other plants and ecosystems by planting it. Countries like Australia would be at major risk of that because of the diversity of plant life is so specialized that it could be easily destroyed if any change to the ecosystem can be too difficult.
Not to mention the fact that in large swathes of land we have up to 1 centimeter to a few millimeters of topsoil, which is not exactly a large volume that can absorb poor crop rotation skills.
But yes, so just to clarify, everything needs to be done in moderation and always be looking towards what the consequences are for trying any new procedure. But that doesn't mean that we should be using that as an excuse not to do anything. So a few people have said, it's too dangerous to do anything on climate change because we might change it in a worse way. It's like, the truth is, I mean, that's absolutely true. We could have some unforeseen effect. The thing is, though, we know climate change is happening. We know it's going to have an effect. So if we do nothing, that's just we're guaranteeing our demise.
This is, in a sense, the human dilemma, where we're able to affect our environment in such a way that every move we make, all the decisions that we make in search of our basic desires, we're pretty likely to have many unseen effects that we probably haven't thought about. We have such capacity to wreak changes across this planet, both for good and bad. It is a lot to expect of people to be able to think about the consequences of all their actions, but in light of modern day, I think it would be too hard for people to increase that by at least a bit.
Yeah. So now we might get a little bit political, because in the end, our governmental bodies are here to solve problems on our behalf. As part of a democracy, we expect that our lawmakers see the concerns of the countries and act in their best interests. Unfortunately, this is not what we're seeing. And a recent thing in the land of Oz has been that There have been student strikes about climate change, protesting the government's inaction, and I support this. I think anything to get awareness going when you can't necessarily directly do anything yourself is really important. The response from lawmakers has been very unfortunate, with a couple, I can't remember the exact quote, but telling them to go back to school, which I think the lawmakers should maybe take their own advice. But this is a very serious issue, and being political on it, it's not a political issue. Climate change should not be a political issue, because that pervades the false idea that politics is an end in itself. Politics is simply a forum which should be a means to finding solutions. It is not simply a debating field for people to figure out how they can make more money. And sorry, this is a big problem with climate change. It's been made, and I'm using air quotes, which you cannot see here, political, for the sake of then being able to argue it without actually looking at what the available evidence is. Any reasonable person looking at the evidence would say, yeah, look, humans are causing climate change. There's still a huge variance in the outcomes that could happen. We need to take these concerns on board and act on it straight away. Unfortunately, the response has been quite short-sighted.
I just want to interject there to tell me the other quote you were thinking about was from another government minister, which was that if children are really committed to stopping climate change, they should stop eating ice cream and eating burgers. I take a few issues with this statement. For one, of course, it's just completely dismissing the issue. For two, of course, they're not accepting that they're in the position of power to deal with the actual carbon dioxide output of our society. It's a bit of a large burden to put on the individual consumer. And also, for another fact, that I think he's not recognizing that a lot of these children are climate activists, and so perhaps many of them are cutting down on their meat consumption.
Well, yeah, the whole thing is he doesn't know about the individual, and I'm sure a lot of them eat ice cream, as I have to admit I do. Yeah.
Rainbow paddle pop.
But one of the things is with this is dismissing the issue isn't going to make it go away ever. Never going to happen. I was going to say something else, but it has eluded me.
Short-term memory loss.
Yeah.
Let's talk about that.
Much like politicians. Especially in Australia. Actually, we have just short-term ministers altogether. No, but when it comes to climate change, it's all about being pragmatic. And the thing is, we look at humans, we look at the human condition. We're not the best individual decision makers and consequentialists. We can't see long term sometimes, and often it is. I mean, it wouldn't be fair. It's fair to say that most humans have a lot of really important issues going on in their immediate lives, dealing with debt, finding a house, anything like that, and that can often make it very difficult to then put aside that and think, what do I do to support the wider world? And that's where government comes in, because what they can do is they have the power to look after and regulate the rules that determine what the whole country is going to do, and so. It is quite a useless thing to be going on and saying all individuals need to do something when you're in a position of power to make sure the rules that are able to sustain our country and sustain our consumerism are put in place effectively.
I think that, yeah, just to, perhaps I would just mention quickly that this climate change, amongst many other issues, is one that can perhaps be curtailed in a sense by greater education. As with, I think me and Matt find that almost any issue can in some way be reduced if people were better educated. And part of that is, of course, if you're listening, don't forget to always educate yourself as well. Institutions will sometimes let us down.
Yeah, so don't forget to always look into things for yourself, but also don't be dogmatic in what you read, because just as we tail off here into the episode, a lot of researching sites such as Google and stuff, fantastic resources, massive innovations in our technological abilities, but their whole way they make money is by keeping you on the site as long as possible. So if that means showing you stuff that you are more likely to read, they will. This doesn't necessarily mean that what you're being shown is incorrect. It just means always take a critical eye to it, and always maybe get someone else's opinion, discuss it with someone, see what seems reasonable.
Yeah, have a little bit of Socratic discussion, such as we like to think we're having here on the show right now.
And with that, we'll wrap up the episode for our first episode. And again, if anyone wants to sponsor us or bribe us, that'll be fine. And hopefully you can join us again if we post another episode, which I hope we will. Until next time.